In this video, we'll start our discussion about DNA and all the associated macromolecules. We're going to start by, first of all, just redefining what DNA is. I know most of you have seen this before in other classes, but it's worth just to remind ourselves that, of course, DNA itself is also known as deoxyribonucleic acid. And it's that double helix that we always hear about. It has a uniform diameter, but it can vary in length. Now, within humans, the DNA comes in 46 molecules, which we consider the chromosomes. And that's what we find in human cells. And of course, the 46 represents 23 pairs. And we'll get more into that. And the DNA and other nucleotides are polymers of, or I should say DNA and the other nucleic acids are polymers of nucleotides. And we'll talk about what a nucleotide is, but before we get there and really look at the structure, let's just kind of say what's part of a nucleotide. So a nucleotide consists of a sugar, also a net. Now, when we're talking about a nucleotide, as we said, for DNA, you remember a nucleotide has to have a sugar, a phosphate, and nitrogenous base. For DNA, the sugar is the deoxyribose sugar. And of course, it does, because DNA is made of nucleotides, it does have that sugar, it does have that phosphate, and it does have a nitrogenous base. Now, the nitrogenous base varies from one nucleotide to the next, and most of you have probably heard of the shorthand, which is A, T, C, and G. Well, these four nucleotides that we find in DNA fall into two groups. One is the purines. The purines, and the hallmark about the purines is that it's a double ring structure, and we'll have a look at it. And under the purines, we have adenine, which is usually written as A, and guanine, which is written as the G. Then we have a second group, and the second group are the pyrimidines. And the pyrimidines differ in that they're not a double ring, but they're a single ring. And here we find cytosine, which is the letter C represented by it, thymine, and then we have, I'm going to change color here just to distinguish it a bit, and then we have uracil. Now, just a note is that when we're looking at DNA, it's cytosine and thymine that are the pyrimidines, whereas when we come to look at RNA, we'll see that thymine is not in RNA, and instead it's replaced by uracil. Now here's just a quick look at a nucleotide, and you can see that it follows the basics that you need for a nucleotide, and that was that you require phosphate, you require sugar, and in this case this is the deoxyribose, and you can tell that's a sugar structure, very typical of a sugar structure. And then we have a nitrogenous base. Now in this case, this nitrogenous base, the way it's put together, represents adenine, and it's really this part that's going to vary between all the letters we just looked at, the A's, the T's, the C's, the G's, etc. Now, let's have a look at the structure. I wouldn't ask you to be able to replicate these on a test, but it's good to get a mental image. And here's the purines. And remember, the purines are a double ring structure. And you can see there's adenine and there's guanine. And you can certainly definitely see the two rings that we're talking about on both of these. And then if we go down to the pyrimidines, cysteine, thymine, and uracil. And once again, you can see it's no longer a double ring structure, but we have a single ring structure. Now, often you hear the shape of DNA described as a double helix, sort of like a spiral staircase. And part of that we refer to as the backbone. And the backbone is the phosphate groups with alternating deoxyribose. And the steps, if you want to think of the steps that make up the spiral staircase, that would actually be the ring-like structures. So either a purine or pyrimidine would be part of that. And here we can see the structure. Here you can see the sort of the structure showing the double ring with the various atoms that are in there. But if you sort of start to simplify it a bit, you can start to see, and this is the way it's often represented, sort of in this very simplified view, with these two backbones. Often we refer to these as part of the backbones. And then in the middle, you can see that we have the nitrogenous bases. You can see adenine, thymine, etc. Now here's a sort of one more like a stick diagram of it. And let's have a look. And what we have here are the phosphates. So you can see the phosphate groups are on the very outside, sort of form the outer perimeters. And then you can see the individual sugars. And remember, there's three parts. There is a phosphate, a sugar, and then the nitrogenous compound. And you can see that here the phosphate and the sugars form almost what looks like a backbone on this side and a backbone for the structure on this side. And often it's referred to that as the sugar phosphate backbone. Now as we look in here, we can see the nitrogenous compounds. And we can see that in DNA, T always interacts with A, and C always interacts with G. So the pairing is always A and T and G and C, or C and G. You can say it either way. Now, one thing to notice is that we always have a pyrimidine and a purine. So every sort of step on the ladder is a combination of one pyrimidine and one purine. And as I said, the pairing is always A with T, and G and C are always together. And you can see some of it here. Is that why? Because you can see these are hydrogen bonds. We have hydrogen bonds forming, and you can see A and T will bond with two of these bonds, and C and G line up in such a way that there's three. So we never find a mix-up between them. It's always AT or C or G. Now, as I said, the nitrogenous bases are held together by hydrogen bonds. And there's always a purine on one strand, and it's always bound to a pyrimidine on the corresponding strand across the way. And as we just said, A and T always come together, and C and G. And A and T are held together by two hydrogen bonds, and C and G by three hydrogen bonds. Now this whole structure, that A always bonds with T and C always bonds with G, gives us something that we call the law of complementary base pairing, which means that if I knew what the letters were on one side of the rung of the ladder, I could predict what the other side would be. Now I just want to talk a bit about more about DNA structure and some basics around it. And let's start with this, a gene. Now our concept of a gene has changed a lot over the last number of decades. In the past, we used to think a gene. Now, a gene is a segment of the DNA that holds instructions how to make RNA, or RNA gets copied from it, and the RNA has instructions how to make the protein. So the flow of information is from a gene to RNA to protein. Now, in the past, we used to always think that one gene would lead to one protein. But we ran into a problem with that idea because there's at least hundreds of thousands of proteins in the body. And then what happened was a project came along called the Human Genome Project, where they started to identify the genes in the human genome. The genome is all of the genes within you. And they came to discover that there were only about numbers are somewhere between 20 and maybe 30,000 genes. So that didn't add up, that if we only had 20,000 genes, and if it was really true that one gene coded for only one protein, that would limit us to that number of proteins, 20,000. We know that there's more than that. So I think the best way to consider a gene now is a gene as a segment of DNA that holds the instructions to make a set of proteins. And we'll see how it does it. It's basically about modifying the RNA copy. Let's get back to this topic of what a genome is. So the genome, you can consider it all the genes within one person. And as I said, we have about 20,000 genes. The numbers vary, somewhere between 20 and 25,000. Now, if I actually look at the strand of DNA, so I'm just going to draw it like it's just a little string here. That's DNA. It's sort of uncoiled. It's not wrapped around histone proteins, which we'll talk about in a bit. If I actually look at sort of the DNA, I notice that I'm going to draw in blue sort of the areas that code for proteins, that the vast majority of the DNA isn't actually directly used to code for a protein. That it's only about 2% of the total DNA is represented by genes, which is basically the area that codes for a protein. Now, that means about 98% of DNA is non-coding. So that means, anything between the gene areas is non-coding. Now, in the past, they used to call it junk DNA, but I think that was inappropriate. I think we called it junk DNA because we didn't quite know what it did and just thought it was all junk. It turns out a lot of this area does have some functions, and it has a role in chromosome structure and also regulation. So roles in chromosome structure and regulates gene activity. Now, it's true that there are some large areas that we cannot assign any function for right now. Maybe we'll discover the functions down the road, or it could just be that truly it is just sort of extra DNA hanging out there. Now, I'm not going to spend too much time here on the history. It's an absolutely fascinating story. But it was, by 1900, they knew DNA. They didn't know it was a double helix. They weren't quite sure what it did, but they knew there was this molecule. And they knew that it contained sugars, phosphates, and also bases. And it wasn't until the 1950s where they started to use a technique called X-ray diffraction that it started to give some ideas as to what the shape was. And in 1962, the Nobel Prize was given to James Watts, Francis Crick, and also Wilkins. There's another person who we should mention in this story, and that was Rosalind Franklin, who did a lot of the early X-ray diffraction work and probably took some of the most important pictures of DNA, which had a very strong influence in finally figuring out the eventual shape of DNA. Sadly, she had died. She had passed away at an early age of 37. And she, of course, the Nobel Prize is only awarded to those who are currently living. So there's quite a story there, and we should acknowledge the role of Rosalind Franklin. Now, just want to mention chromatin, because there's a lot of names. You're just going to start here: chromosomes, chromatin, DNA. And really, what is chromatin? When I'm talking about chromatin, I'm talking about DNA, but the DNA is mixed. It's complexed with very important proteins, so it's complex with proteins. And as I said earlier, DNA comes in sort of chunks. This chromatin is packaged in chunks, and these chunks are the 46 chromosomes that we find in most of our cells. It's about 2 meters long if you actually added it all up and stretched it all out. Now, unfortunately, or maybe fortunately, in a non-dividing cell, the chromatin is so thin you can't see it with a light microscope. And even under an electron microscope, it just looks sort of granular. Let's have a look here. This tries to present the sort of organization of DNA. Because often what happens is we're used to looking at DNA in this structure, sort of that twisted staircase, double helix, ladder. And then at other times we're shown pictures of this and this and this do not look the same. If you want sort of an analogous sort of thought, it's very much kind of like protein, where we went from primary to secondary to tertiary to quaternary structure, where there's folding and there's sort of changes. So let's have a look. When we're looking at DNA like this, what are we looking at? We're looking at the sugar phosphate backbones. That's the sugar phosphate backbone right there. And then we're looking at the basically nitrogenous bases, so the A's, the T's, the C's, and the G's. Now, most of the time in the cell, DNA is not hanging out like this. Most DNA is not. That the DNA gets packaged. And then what we have are these things, I'm going to zoom in, called histones. And histones are a series of proteins. In this case, they kind of look like hockey pucks. And what happens is the DNA wraps around the histones. And this does a number of things. And one of the things it can do, it can help to regulate whether that part of the DNA is red or not red or available for protein synthesis. And then you can see, so it's almost like the first order is just the double helix, and then sort of the second type of packaging is we start to wrap around histone proteins. And then what happens is we start to, once the DNA, that's the DNA there, is wrapped around the histone proteins, then the histone proteins start to sort of align themselves almost like an accordion, almost like a bit of a folding. And that will also shorten the length. And then the whole thing tends to fold back on itself. So it's very much like an origami that's occurring. So at first, we have our DNA double helix, and then the DNA winds around the histones, and they form something called core particles. This is a core particle right there. And then the chromatin folds into sort of a zigzag type fiber. And then the fiber folds even more into these irregular loops. So what we're doing is taking what's a very, very, very long, in a cellular molecular way, very long molecule, and basically packaging it to be a shorter, and also associating it with these various proteins. Now, let's talk a bit more about the histones. The histone proteins, these are absolutely critical for DNA packaging, and they come in clusters. It kind of looks like it's just one, but the histone clusters in groups of 8 molecules together, and the DNA winds around it. Kind of, if you want an analogy, it's like thread around a spool. And the chromatin consists of thousands of these repeating nucleosomes. Now, what's a nucleosome? This is a new term we just sort of introduced right here, the nucleosome. So the nucleosome, let's change this out, get a different color here. The nucleosome is basically the core particle, and the core particle is the histone with the DNA, and it goes around about 1.5 times. And then there is a little segment of DNA called the linker. So that's the section that connects one core particle to the next. And let me back up so we can walk through this on the diagram. So here we have the DNA. That's the DNA right there, right? And here we have the bundle of 8 histone proteins. The DNA wraps around it. So this one little section, let me zoom in. So this one little section here, which is the DNA and the histone proteins, that's the core particle. The area between core particles is the linker. So here's a core particle, right? That's a core particle. That's a core particle. And of course, these are the linker parts. As we said, one nucleosome is a core particle with the linker. Now, once I've started to package it, and it will reduce quite a bit in size, that the chromatin forms a complex, the irregular loops, it coils even, and by the time you're done, it's about 1,000 times shorter than the original molecule that you started with. Now we're going to end there, and then we're going to pick up the next video. We're going to come back and talk about chromatin and the chromosomes a bit more. Nitrogenous waste.